that, we have thought about idea not establishing real-time PCR lab, but having what's called mobile suitcase laboratory. And it's as it stands, it's a suitcase laboratory. It's actually like this. It's composed of power pack, solar panel, and um, uh, uh, and these suitcases for the detection and extraction. As you see from this slide, we have one suitcase lab where you do your sample preparation, and then one suitcase lab where you can do detection and amplification, but not using PCR. I will tell you which method in a minute. Of course, if you ask yourself, okay, if I am working with a pathogen like S3 pathogen or S4 pathogen or something very highly pathogenic like coronavirus, or like we use it sometimes for Ebola virus in Africa. So in this case, we use what's called the glove box. Glove box is a closed system where inside it you are sure that you inactivate the sample while you are working. For example, in the agriculture or in the veterinary science, avian influenza. Avian influenza is a virus can infect human. For that, you need to be sure that you inactivate the virus while you are working and avoiding that you get the infection. Therefore, you can do sample inactivation inside this glove box. Why we need a suitcase lab? As you see, all the machines working here, it's sitting on a foam layer, so you can transport it, and you are not afraid that the machines will break. We can even drop it from a helicopter to rural area. And also, you see, everything is sealed. So if you do any working here on the surface, will not contaminate the underneath foam layer because everything is sealed. And we have used, instead of using disinfection solution, we have used disinfection wipes. And this is very important because if you have a solution inside the suitcase lab, the problem is very big, because if the fluid open, it will destroy the machine. Because of that, we have used instead of that wipes, and we have used this discarder in order to get rid of all the waste and inactivated while working using 2% bleach. The most important thing, it's a molecular diagnostic method, but all reagent we are using is lyophilized. That means you can put it at room temperature without any problem. So no need for freezer. And this is a good advantage of this technology. And which method we have used in this suitcase lab? There is a called isothermal amplification technology. So instead of using real-time PCR, which is called polymerase chain reaction, which you need 3 temperature, we can work only at 1 temperature. And as you see, there are many methods. Most known example in science is LAMP, where you can amplify at 60-65 degree for 30 minutes. But we have selected the recombinase polymerase amplification assay, where you can run the assay at 40 degree, and you can get a results in 3 to 10 minutes, and you have 2 primer, and you have a probe system. If you compare the loop-mediated isothermal amplification, the LAMP, and the recombinase polymerase amplification, as you see, both are isothermal. Both you can do visual readout, or you use portable machine. But of course, the RBA, you can use only 2 primers, while in the lab you need to have 6 primers. In RBA, it's a simple assay design, because if you have 2 primers, it's easy to design. But in the LAMP, it's not easy because it's 6 pair of primers. Of course, the RBA has an advantage. You can store it at room temperature without having any problem. I will tell you here what is the recombinase polymerase amplification. And I will show you now a video how this amplification works in comparison to the real-time PCR. As you see, we have two primer, one forward, one reverse primer. You have what's called the recombinase. This recombinase will bind to the primer and make a complex which at the end invade the double strand DNA. So this recombinase primer complex will just invade the double strand DNA and go inside the double strand DNA. And as you see, it will be amplified and going on and on and on. And this is how this recombinase polymerase amplification works. You don't need to use different temperature, but instead you use enzyme and protein to do the amplification. The good thing about this RBA is it uses also a probe system. It's different from real-time PCR probe because you have around 30 bases to the 5' end, 15 bases to the 3' end, fluorophore quencher, and you have tetrahydrofuran residue. Once this is bind to the target, then the exonuclease will come and make a cut, and the quencher will be separated from the fluorescent, and you see a fluorescent signal on your screen. And this is how the RPA assay, the recombinase polymerase amplification. I know that most of you know the PCR, but here the RPA assay is very fast. You can have this amplification and detection in around 10 minutes. We have used also a novel extraction because you see, if you are working in the lab, usually extraction is a horrible thing to do, and it takes several hours to do it. But what we have done, we have together with QIAGEN, we have developed this rapid extraction protocol where you can do rapid extraction of nucleic acid. Usually pathogen is inside the cell, as you see here. And what's happened is that we put on the sample a lysis buffer, a magnetic beads, and heat everything for 95 degrees for 10 minutes. That means you put your sample, you put lysis buffer, magnetic beads, and cook it. After 15 or 10 minutes, you see that the DNA, RNA will be in the fluid, but the magnetic beads will capture the inhibitors. And if you put a magnet, the magnetic beads will take all the inhibitors, and you have your DNA free in the fluid where you can take it out and go to the next level. So in here, we call it reverse purification. And instead of purifying the DNA or RNA, we purify we capture the inhibitors like proteins, cell membranes, and other stuff, and keep the DNA and RNA free in the fluid. I will give you here an example of two. Of course, we have used it for many pathogens, including coronavirus. But I will give you two examples, which very important example for agriculture field, which is Mycobacterium avium subspecies paratuberculosis or lumpy skin disease virus. Of course, all of you know the MAP, the Mycobacterium avium subspecies paratuberculosis, which cause a chronic diarrhea. It's a wasting disease and affecting mainly the old cows. And it's a very nasty disease which can lead to a highly economic losses. On the other hand, lumpy skin disease also is known with these lumps on the skin. So these nodules is the skin which open up and do this nasty skin and generalized disease in cattle also. First thing we do when develop these assays, we determine how many copies I can detect, how many copies from the virus I can, or from the bacteria I can detect. And as you see here, in case of MAP, we can detect around 1 copy in 8 minutes. This is very, very fast. 1 copy, if you would like to detect it in real-time PCR, it takes almost 2 hours. Here we can detect down to 10 copies in around 11 minutes. You see, this is the difference between a virus and a bacteria. But still, you can detect around 10 copies in less than 15 minutes, which is very fast if you compare it to the real-time PCR. If you combine, the second thing is we always ask ourselves, is my assay very specific? And we need to do specificity test in case of paratuberculosis. We do with many mycobacterium species and also other bacteria. And as you see, our assay can detect only the paratuberculosis, but not other bacteria. The lambskin disease, it detects lambskin, sheeppox, and goatpox. All the three are three viruses belong to the same viral family. And in the second part of my presentation, I will tell you how you can differentiate between them. But at the PCR level and at our RPA level, you cannot differentiate. You can detect these three viruses at the same time. We need also to study with clinical sample, and as you see here, we have real-time PCR in comparison with RPA. You see that you have a sample from cycle 20 to cycle 35, and all of them you can detect almost 90% of this sample in less than 15 minutes in RPA, while the lambskin disease, as you see, you can detect all sample, whatever is CT up to 38, you can detect it in around 5 minutes. So it's a very fast technology where you can detect your target in very, very fast. And this is really a revolution here. So what we have used, since we are in the agriculture, so I show you two examples from animal viruses, but these technology actually work exactly like the real-time PCR. And since we are in the molecular genetic department, it's very important to show you we have used also the same technology to detect animal species in the meat. So usually, of course, this is photos from Riva in Germany, but you see you have many type of meat, either in open an open fridge for selling, or either bagged in a vacuum bag. And all this one they tell you it's a minced meat or this kind of meat, and it's the source maybe is from cow, from pork, from horse, and so on. And what we have done is that we said, can we use this technology and use the genetic material in the meat to differentiate between the meat, to detect which meat source is that? Do I have pork in my meat? Do I have horse in my meat? Do I have other animal I don't want to mention, because we all know all the contamination the people in the open market can put it inside it. We have used the 6 animal: turkey, chicken, sheep, cow, horse, and pork, because of it's important to Germany. Of course, if I am working in Egypt, I will put also another donkey, hot donkey, and dog. And what else we can expect? I think a lot of other species can be expected in the meat on our streets. So we can detect, as you see, you can detect very few copies, from 1 copy to 30 copies, and in less than 11 minutes. So all these species can be detected simultaneously, very efficiently, and also in less than 11 minutes. As you see, the assay again is very specific. Horse detecting only horse, pork is pork, chicken, turkey is turkey, beef is beef, and sheep is sheep. That means you can detect very specifically your target animal in the meat. And by this, we can identify as our meat contain only one animal or several animals, and so on. This part, of course, has been done by a PhD student in my group, Jonas, and my technician, Susanna. So this part, which I spoke with you about, the suitcase lab, we have developed a mobile suitcase lab. You can take it anywhere. It's work with solar panel and power pack. You can use a novel technology called recombinase polymerase amplification assay, and we can do amplification, extraction, and detection in a maximum of 30 minutes without any problem. That means extraction and detection both in 30 minutes. And if you compare it to the real-time PCR, this is very fast. This, I will end my first part of the presentation, and I will move to my second part of the presentation, how to use sequencing at point of need. I told you how to use molecular assay similar to the PCR in the field, which is the RPA. But now I will take you to another technology, and this is called nanopore sequencing. And the idea of this sequencing technology is very simple. I will operate the video here. As you see, you have a small pore here, and whenever something comes through this pore, it will change the current. You see, another one will change the current, and so on. And by this, you can sequence the DNA via grasping the DNA through this pore. And while changing the current, you can see it's G, T, C, and so on. So it's a very nice technology where you can do sequencing directly at point of care, and also without doing any amplification. You just put the DNA as it is, and it will grasp through this pore, as you see, and the change in the current will be recorded, and you can really see which sequence in real time. And this is very important, very good thing about this technology. The other important thing about this technology is that the device we use it also inside the suitcase lab, but it's very small. As you see, this is our sequencing machine, even smaller than the laptop or even everything. But of course, you need everything to do library preparation. And as I told you, you just grasp the DNA from the pore, and by changing in the current coming this direction, you will see that there is a change in the current, and it will be recorded as G, T, C, A, and so on. And we have, if you compare this, of course, with the other big player in sequencing, you have many people, many companies produce sequencing. The most great example is Illumina, but it's very expensive. But here is a very simple, cheap lab-on-chip method. So I will tell you two examples from also the veterinary medicine where sequencing is needed. We have two viruses. One is a foot-and-mouth disease virus, which is doing a big problem in Egypt, and also Cabri-Pox virus. This is an RNA virus. It's 8,000 bases. Here, Cabri-Pox, it's a pox virus, and it is around 150,000 bases. The foot-and-mouth disease has 7 distinct serotypes. You have 7 serotypes for this virus. While Cabri-Pox, you have a host-specific sheep-pox, goat-pox, and lumpy skin disease. The idea is that foot-and-mouth disease virus, you cannot differentiate between the 7 types using PCR. It will not happen. It will not work. They are very close, and sequencing is the only method to differentiate between them. The same is true for Cabri-Pox virus. Sequencing is the only method to differentiate. That means here there is a PCR limitation. You cannot differentiate using PCR, and you need to do sequencing. And this is what we have done. We have used the sequencing to serotype the foot-and-mouth disease virus and differentiate between Cabri-Pox virus. As I told you, in Germany, foot-and-mouth disease virus is 4 lab, and you need to have, of course, do the inactivation into the glove box and do everything else on the suitcase lab. What I would like to tell you here also, it takes almost 7 hours to extract prepare the lab. It's not very fast like the RPA, but still, you need a lot of time to prepare your sample for sequencing because you need highly purified DNA. For the this is for RNA viruses, because foot-and-mouth disease is an RNA virus, but here is a Cabri-Pox virus, and this is a DNA, and sequencing for DNA is very fast. From extraction to detection is around 2.5 hours. The most important thing is that we can do offline blast search using Genius program in around 5 minutes. And this is the very important point here, because usually if you do sequencing, you need to do blast search online. But if you do that, you will have a big problem, because you need a stable connection, you have a powerful online version, and so on. But here in this program, using Genius, you can do offline blast search, and you don't need the online version where you will have big trouble or problem with it. As you see, in foot and mouth disease virus, we have 70 serotypes. And what we have done, we said, okay, should we rely on whole genome, or should we rely on a particular gene region? And as you see, in this case, in foot and mouth disease virus, the B1 give a very good results, even more better than the whole genome. So in this case, B1 is a target gene in order to detect this virus using sequencing. Why? Because this gene is responsible for the production of the specific outer protein of the virus. And it's not surprising that this region is highly specific for each serotypes. In case of Capri box virus, as I told you, we have lambskin, gold box, and sheep box, and we have investigated also a whole genome and also a targeted gene. In this case, in a DNA level, both whole genome and target gene produce a very good specificity and can identify correctly the Capri box viruses. So in summary, I can tell you here, we were able to serotype foot and mouth disease virus and differentiate between Capri box viruses using sequencing, and not any sequencing, it's a nanopore sequencing, where you can do this stuff directly on site without having any problem, and with having high efficiently as you do it in the lab, but at the field level. Of course, I would like to thank in this part all my colleagues in Egypt, especially Professor Shalabi, who supported a lot of this work. So in less than 5 hours, you can do sequencing. No need for bioinformatic background, and I'm sure you will be surprised to hear that, but it's a user-friendly programming using Genius, where you can do rapid data analysis. You see the sequencing, it's the good thing about also this sequencing method, besides you can do it in the suitcase lab and very mobile, it's cost you almost to buy the machine 1,000 euro. So for buying a machine, to buy a sequencer, you need 100,000 euro, but this is only 1,000 euro. You don't need an internet connection. The product side is unlimited. So if you do normal sequencing, you do up to 1,000 bases, but here you can do up to 5,000 bases. We have colleagues succeeded to have a DNA sequence up to 1 million bases. So it's a very nice technology where it's open up for many stuff in your work. I will stop my presentation here. I finished the presentation early in order to get questions from your side. Please, I'm speaking Arabic. If you would like to ask in Arabic, please ask, and I will hope to answer it. But I would like to tell you what's my overall summary here. What's my overall summary here? The Scottish biologists say, think globally and act locally. So nowadays, he said this almost 80 years back or something like that. And as you know now, that this is very important, especially in the Corona time and other time. You need to act locally and develop yourself, and what you are doing will affect the whole globe and will affect the other human being living in other country of the world. Thanks for attention, and I'm ready for questions.
Thank you, Dr. Ahmed, for this interesting lectures. And now we open questions. So please, if you have any question, please leave your question in the chat area. So I have just one question, Dr. Ahmed, for this. Um, are you working now for developing more kits for detecting infectious disease, more infectious disease, or just you stopped at this level?
No. We have developed more than around 30 pathogens. I just show examples from veterinary field, but we have developed it for many pathogens, for Salmonella, for E. coli, for many viruses like the coronavirus, Ebola virus, dengue, chikungunya, and other viruses which affect human and other zoonotic diseases also. So we have a long list of pathogen. We have developed the technology for it.
Oh, perfect. And how they are available to Egypt, or can anyone buy these kits? Or
Yeah. It's like exactly like the PCR. You buy the basic kits
okay,
from the company, and you buy the primer and probe from another company. So
okay,
it's exactly like real-time PCR.
okay. We have just one question here. What are the regions you targeted to do pathogen specificity test?
Okay. No, there are two different things here. There are two different things. When we develop an assay, we select a conserved region. We know that this region is conserved for a specific pathogen. This is what we select the region, yeah. And you can do, of course, plus search for your primer using the NCBI in order to see if your primer are specific or not. But what we will do is that we have sample containing other pathogen, and then we test our primer against this pathogen.
Okay. And another question is this kit expensive?
It's exactly the same price like PCR. In our hand here, it costs us around 4 to 6 euro per sample.
Okay, 6 euro.
So that's mean 100 Egyptian pound.
Okay, another question. Also, if you please tell us about practical steps for isothermal amplification. So I think you described this in the lecture.
No, Dr. Nagwa, what you mean by practical steps?
Uh, I think she means uh, the basic steps of thermal amplification for PCR. Or
What I can do, let me do it because we have time, yeah. So guys,
yes,
what I will do is that I will show you a video how we do the test directly in the field. But this is of course for coronavirus, yeah.
okay,
So I can show you this video so you can see how exactly we do it in real time. If this is good for you
yes, you
in the
can.
laboratory. Okay, so it's around I think 15 minutes video, but it will show you exactly to be aware how we do. Just a minute, I need to open my video here. So sorry. Bum, bum, bum. Oh, I hate this. So I will show you a video how to do this, but let me test first that everything is working. This is Tonga, this is a very nice question, so we can explain more how the test is done. Do you see my screen?
Yeah.
Do you see
Yes.
the video running?
Yes.
Okay, now it will run a video for around 15 minutes to explain to you how we work. Hi, everybody. I will tell you first what you need to have from equipment in order to perform the assay. As you see, we have a heat block adjusted at 90 degrees. We have a vortex. We have a centrifuge. We have a rack which can take 0.2, 0.5, and 1.5 or 2 ml. We have the tips 10, 100, 1000, and we have also the pipette 1000, 110, and then we have the gloves, disinfection wipes, the kits which we'll use for performing the assay, and the most important part is the tube scanner. Here we will start with preparing the extraction kits. As you see here, we have the Swift swap extraction kits, and first we need to activate the enzyme which come with the kits. So we will put 1 ml of component A, so A in German, sorry, into component C. This is the first step you will do it. This gives you also a feeling about working in the suitcase lab, how it look like. You pipette up and down in order to mix the component and to assure that the pellet is completely dissolved. Then you include this 1 ml back to the component E. You mix by inversion in order to ensure that the component C is completely mixed or into the component E. If you would like to do it for swap, we usually use 500 microliter and pipette it into 2 ml Eppendorf tube. So directly, when you collect the nasal swab or buccal swab, then what you need to do is that you can directly put the swab into the lysis buffer. As you see, the swab is directly into the lysis buffer. This is how you do inactivation of the virus if it exists while you are working. And this is, you put it directly into a heat block for 10 minutes. During these 10 minutes, you can prepare your mixes of the RAA reaction. For the REA, you will have several important steps. You have the buffer, which contains the rehydration buffer. You will have the magnesium acetate, and you will have the reaction positive control. In REA, you will have a pellet which contain all the reagent you need to start the reaction, including RT enzyme, including primer and proof, and also including the enzyme and protein which operate the RAA reaction. We always have one tube for the sample, one tube for the positive control, and one tube for the negative control. And you need to pipette everything you need into the lid of the tubes. First, you put 40 microliter of rehydration buffer into the lid. Then you pipette 5 microliter of the magnesium again into the lid. Of course, if you would like to make several samples, you can still prepare a master mix and pipette it directly into the lid. But since you are working only with one sample, one negative and one positive, we will just do it individually in each lid, that you know exactly what we put into the lid. The magnesium is very important because it acts as a catalyst to start the reaction. We don't pipette directly into the billet, because if you pipette this stuff into the tubes, the problem is that the reaction will start immediately. For that, it's very important to be sure that you pipette everything into the lid. This is a positive control. We have also a negative control. We need to wait for the 10 minutes until the lysis is already over, and then we pipette 5 microliter of the sample into the last lid. During this, I will show you how to adjust the machine. So, the tube scanner is very important. It's come with a lock. And this lock here, you need to be sure it's open, because other than this, it will make a problem for the detector. Because you see, I need to make this open, and you see, you will hear that the fluorescent is moving. And this is very important. If you don't do that, the detector will not move and you have a problem. For that, it's very important to unlock this first. After that, you open by pressing long on this button. It will show you the sign. It will make self-testing to assure that everything is working. اسمعوا. You need to press New Test and select the program. And this is the RA COVID-19 test. And then you have 3 tubes. And this is the 3 tubes you will use in this reaction. So you press Start. It tells you if these are the tubes. You tell them yes. And until we confirm that all tubes are inside and start run. And this is the next step. After 10 minutes, you are sure that the virus is inactivated and extracted if it exists. Be sure to mix and centrifuge to avoid that any remnant is already in the lid. Then open it, take 5 microliter and put it directly into the lid. After that, you close the lid. You need to mark all the tube, which is positive, which is negative, and what's the sample number. Then you do what's called SMS, spin, mix, spin. So you need to be sure that the fluid all come down and touch the pellet. Then you need to be sure that everything is mixed. Very good. And now it's time to insert everything into the tube scanner. The tube, now you press the green, and as you see, the test will start. The machine will be first heat up, as you see here. We reach a temperature 39 degrees as we wanted, and then for up to 15 minutes. So the reaction will start, or the preheating started already, and after 4 minutes, it will open up to mix. After 4 minutes, it will tell you, please open the lid, mix and close the lid. So now we will open the lid, take the tube out, and this time we'll do mix and spin. It's very important to do this step because the RAA reaction is working in small colloids, and you need to break these colloids in order to have a very good mixed fluid and the primer come in contact with your sample. Once you close the lid, it will start automatically, and now you hear the fluorescent signal or the movement of the detector, each 30 seconds, it go and do this FAM, the detection of FAM fluorescent in each tube. During this time, you can press on the tube name to see the results. And of course, since we have no curves, there is nothing is on the screen. You see, this is the start of the signal. It's really at the beginning, nothing happening. It's clear negative. So let us wait for another 3 minutes and look to the results again. As you see, after a few minutes, if we press into the results bar, you see this is our sample. As you see, a very good amplification of the curve up to 600. If you see now the positive control, so this sample is positive. If you see our positive control, also a very good signal coming, Alice amplification curve. And if you see the negative control, you will understand the difference now. You see, this is a flat curve at 60 millivolt, which means this is a negative. So I think you now have a feeling exactly how the assay is working from the beginning till the end in real time. Everything will maybe in less than 20 or 30 minutes.
Actually, Dr. Ahmed, this is a very, very interesting video. Thank you for showing this to us.
Yeah.
Thank you. We have a question from which organism PRA has been extracted?
In the video or generally?
Generally.
You can use it as I thought. We have developed it for almost 30 pathogen. Let me bring, I have a list here, so maybe we'll have a clue which pathogen we have used for it. Yeah, please go to the next question until I found the list.
Okay. Okay, we have a question. Does PRA technique works with different types of microorganisms, bacteria, fungi, in addition to viruses, as a rapid detection tool?
Yes, yes, we are. I'm a virologist. Because of that, I show you many viruses, but we have used it for parasites, fungi, bacteria, and everything. And as I show you also in the video, we have used it to determine the animal species using the DNA in the meat. So anything which work at the DNA and RNA, it will work. We have also friends have used this for cancer detection, the mutation in cancer. So it's exactly work like the PCR, the same technology, but instead of having these three temperatures, it's one temperature, and you can have amplification in less than 10 minutes.
Okay. Is this video is available? Can we get the link of this video? Because some attendees are interested to have the video.
So the video, we are waiting to have all the approval from the company's name mentioned in the video, because they are always very, very restrictive when it's come to stuff online. But I have two other videos, or one video. I can show you this one video online. Just a minute. I will
On YouTube channel or
on YouTube. I will send the link in the chat. So in the chat, there will be a YouTube link, and you can have a video on the YouTube.
Okay.
So I have just put it in the chat region. So what would we propose? So any other questions?
I think we don't have other question for that. Okay. Any questions? Okay.
You can ask in Arabic, guys. There's no problem at all. So.
Okay. So I think how much case and device for how much cost? I think how much kits and device for isothermal amplification. I mean the cost of the everything, the kits, the mobile.
The suitcase lab. The suitcase lab is
Yes.
around. The suitcase lab with all the small devices is 2,000 euro. The detection device alone from Kaizen, it can cost 4,000 euro alone. So that means you need calculate around 6 to 7,000 euro, 140,000 Egyptian pound. But of course, it's cheaper than the PCR machines.
Okay. So
Yeah,
just to regarding to the source of PRA, so
RPA. Yeah.
yeah, RPA. So in other words, we know the source of tag polymerase. What is the source of RPA? Sorry, RPA.
Combination, yeah, the tech mode. You
Yeah.
have a mix of
Yeah.
enzyme and protein, which is if you write recombinase polymerase amplification, it will bring you the original manuscript from 2006, where it used phage enzyme UVX. It's a recombinase UVXY UVXX. This is the recombinase part. You have what's called GB32. It's an single strand binding protein, and we have a polymerase from Bacillus subtilis, which can be used in RPA reaction.
Okay, we have another questions. What is the difference between loop-mediated isothermal amplification and RPA?
Sorry, what the difference between what?
Between loop-mediated isothermal
Loop mediated.
amplification and
Okay.
RPA.
I think you came very late in my lecture because I already showed that. But
Okay.
no, but I can show it again. So the difference I have here a table where I can show the difference between loop-mediated hydrothermal amplification technology and RBA. So I will share screen again. So as you see from this, loop-mediated, you need 6 primers. RBA only 2 primers. Both you can do visual readout. Lamp use 65-degree, RPA use 39 to 40-degree. Both can easy to implement it in field. The lamp you need 30 minutes to have to read the results. RBA maximum 15 minutes. RBA only 2 primers. This is make the assay design is very easy. In RBA we have a probe system like the Techman probe. We have what's called exo probe. So far the agent of RBA can store at room temperature while lamp not. This is the difference between RPA and lamp.
Okay. So I now okay. Thank you, Dr. Ahmed Abdelwahed, about the declaration about the PRA source. Could you please send the paper link or title? Thanks in advance. Do you have of course you have a publication about this suitcase.
Okay, the
Yeah.
publication about the suitcase. I will send it in the chat
Or
also. Yeah.
or you can, I think you can provide your ResearchGate link also, so people may be interested to know this.
Yeah. If they write my name, but let me. I will send the suitcase lab, the original manuscript about the suitcase lab, that everybody read about it. So here is the original, and I will also uh, send a review article about different technology used in the field at the field level.
Okay.
I will also put this in the chat part.
Okay. Yes. So I think there is no more questions. I would like to thank you again. I think everybody in this chat enjoyed with this interesting lecture. Thank you, Dr. Ahmed, for being with us in MGBS, and hope to see you next year, inshallah.
Inshallah. And collaborate with everybody from you.
Thank you. Thank you,
Okay.
Dr. Ahmed. Okay.
All the welcome. Many
Good.
thanks. Many thanks, Ahmed. All the best.
Okay.
All
Bye.
the best.
Bye.
Bye-bye.
Okay. We will provide the link for registration. So if anyone missed the registration, we will leave it for 10 minutes and then we will end the meeting. Thank you so much, and see you next week in new lectures. Goodbye. السلام عليكم زملائي الأعزاء. بالنسبة للشهادة الحضور، جالنا إيميلات كتير جدا بالنسبة للشهادة. يعني احنا النظام بتاعنا مختلف، احنا مش هينفع نطلع شهادة بكل محاضرة عشان حضراتكم الوقت والمجهود وكده، ان مفيش حد في التيم عنده وقت ان احنا نعمل شهادة كل محاضرة. فاحنا حضراتكم هنطلع شهادة واحدة بعد انتهاء السيمينارات. احنا بس بنسجل من خلال الإيميل، وبعد كده ان شاء الله هيكون في ابلكيشن خاص بالشهادة، فيه هيبقى الاسم والكلية واليونيفرسيتي وكل حاجة. فموضوع الشهادات ده احنا كتبناه، بنكتبه حتى في يعني تحت كل ايميل ان هيتم اصدار شهادة واحدة في نهاية السيمينارات. فده احنا كاتبين في الملاحظات، بس الواضح ان في ناس ممكن ما بتاخدش بالها، فعشان كده بيجي لنا ايميلات كتير وبنسأل الشهادة حضراتكم هنطلع شهادة بحضور جميع السيمينارات في اخر. ان شاء الله بعد ما ننتهي من جميع السيمينارات هنعمل ابلكيشن خاص، ونبتدي ان احنا ايه نشوف مين اللي حضر معانا من خلال تسجيل كل محاضرة، وهنطلع له شهادة وهنبعتها له طبعا عشان تبقى مختومة، هتتبعت بالبريد السريع والقصة دي كلها. وان شاء الله كل التفاصيل والحاجات دي هتبقى في ايميل لوحدها. شكرا جزيلا.